Hello students, today we will discuss about the topic heredity. In heredity, I would like to discuss about Mendel. As you know, he is also known as father of genetics. Now the question arises in your mind why he is known as father of genetics. Although the genetics work is already given by various scientists, there are some different scientists who work on genetics, heredity, but they were not so successful as Mendel. So actually the first term or concept comes in your mind, that is what actually Mendel do, which is very far away from that time. First, about the history of Mendel. He was born on 22nd July 1822 in a German family in Hansendorf village. In Silesia city, Austria. Exactly, he was born on 22nd July 1822 in German family in Hangendorf village, which is situated in Silesia city and in Austria. At that time, that Austria was known as Czechoslovakia. His father was a farmer, so he belonged to a very poor family. His earlier studies happened in the same village. Later on, he moved towards Broome, where he entered as Faculty of Philosophy in the Department of Natural History and Agriculture, where he was influenced by the work of Karl Nestle. He was actually working on hereditary traits of plants and animals, especially sheep. There he completed his there he completed his MSc, and in year 1843 he began his training as priest. Where in his spare time, as I told you earlier, he is very influenced by Karl Nessler. So he want to utilize his spare time, and there he work on pea plant in the garden of church. He selected pea plant, garden pea, and work on that. This is also known as Python satire. He started work on that in year 1856, and later on he completed his work in year 1864. So you can realize that he did his work approximate up to 8 years. After completing his work in year 1864, he submitted his experiments data conclusion to Brune Natural History Society in year 1865, and that was published in the annual magazine of Natural History Society in year 1866. As hybridization experiment on plants. But that was the bad luck of Mendel, that nobody can analyze what he has done. Nobody can understand because the work was far away from that time. Now, few questions that are usually asked at the CBSE exams. These are: first one, why he chose pea plant for his experiment. Now I'm going to tell the answer of this. Because the first one, pea plant contain various distinguished features which can be easily observed by naked eyes. Now I'm going to write the reasons. Like first one, it contains various variety. You can choose any variety here. Next one, it's easily available. Next, it can be grown very easily in every kind of soil, and it has a very short lifespan of 6 months. Next, it contains bisexual flower in which self and cross pollination can be done easily. Next, as I told you earlier, it contains various distinguished features which can be observed by naked eyes easily. It contains approximately 39 distinguished features, and from those 39 distinct features, Mendel selects 7 features. It can produce high variety of seeds, so there is no limitation for the next generation. So these are some basic fundamental reason why he chose pea plant. Now the next question that usually comes, what the traits are selected by Mendel? As I told you earlier, although 39 distinct features are available, but Mendel chose 7 characters from them, which are actually situated on 4 different chromosomes that was later on discovered by Blixt. Actually, he was so lucky that he chose those characters which are found on different chromosomes. Now I'm going to tell you about the traits selected by Mendel and also tell about the dominant and recessive feature of those traits. So first of all, I want to write characters here chosen by Mendel, and later on I will write about dominant and recessive. Let me start the first character. The first character was stem height. The dominant feature is tall here. Either a plant could be a tall plant or a short plant. The tallness feature is the dominant feature there, so usually this one is expressed as capital T. As I told you earlier that the dominant feature is always should be written in capital letter, while the recessive feature it's small t, shortness. Now the second feature is flower color. That was violet or white. So violet or colored flower is the dominant feature, while white is the recessive factor. You can express this as capital V or small v. Flower position. Either the flower found at the axial point or terminal point. Axial on axis or away from the axis at the any branch terminal. It's expressed as capital A and small a. Now the fourth feature is pod shape. Its fruit is known as pod. So pod shape, either this one is found in the flatted condition or constraint. The next feature is pod color. Green is the dominant one and yellow is the recessive. The next one is seed shape. It could be round or wrinkled. Round is the dominant feature, so expressed as capital R, and wrinkled is the recessive feature, so expressed as small r. The next feature is seed color. Here, yellow is the dominant feature, while green is the recessive. So these are some specific characters which are studied by Mendel. Now the third question that is usually asked, that was what kind of techniques used by Mendel for his experiments. So right now I am going to tell about the techniques used by Mendel. For his hybridization experiments on pea plant. He used two main techniques. The first one is called as emasculation. Emasculation means removal of stamen from the flower in the bud state. By doing this process, he actually wants to stop self-pollination. By removal of all the stamen in the bud state, he actually removes all the male part of flower. And because of that, pollen grains are not available to germinate there or to do pollination there. So first of all, he stopped pollination, and later on he also introduced some specific kind of pollen grain of his desire to the plant. Now the second main problem is if he wants to spread this desirable pollen grain on that, so how he can be ensured that it is fertilized by those pollen grain which are spreaded by him, or this one is fertilized by some other pollen grain. So to ensure this, he again do a specific exercise, or he used a specific technique known as bagging. It's a very simple technique in which he cover up that emasculated flower with the help of a paper bag. Now question arises, ma'am, why he choose paper bag? Why not a polythene bag? Remember student, plant part can breathe itself. If there is a flower, it can breathe itself. If you cover that flower with the help of a polythene bag, how it will exchange the air, how it can respire there, so it will not survive. That's why he cover up that emasculated flower with the help of a paper bag. This is called as bagging, and this one is done to stop cross pollination. After using these two techniques, he also introduced a specific branch of science that is called as biometrics or statistics. He applied maths in biology first time, in which whatever experiments are done, he maintained them individual. That means if the first experiment on, suppose the first experiment on plant's height, so he make a data of that, he make a record of that, and in which each and every data was totally isolated from other experiment, and later on he calculated overall data in a very simple manner. Although although the ratio was too typical, like I can explain this. Suppose you know about the Mendelian ratio, that is 3:1 or 1:2:1. But exactly the calculation was not like that. The calculation was, suppose he has taken a field in which 1,000 plants are grown, and he calculated out of 1,000 to 7 to 6 plants are like tall plant, and rest of the plants were short plants, and the ratio was like just like this 2.9345 ratio. Rest the point. Um, Later on, the most simplest conclusion is derived with the help of this biometric branch. So, at the last, he concluded that instead of saying 2.9345 like digit, you should pronounce that approximate 3:1 plants look like the 3 in the 3:1, 3 stands for the tall plant and 1 for dwarf plant. So exactly he applied a simple method called as biometrics, and because of that his conclusion were error free. Uh, as I earlier told you that in spite of Mendel, some other scientists were also working on hybridization experiments, but they were not successful. This is the main reason behind it, biometrics. Now, next point is why Mendel is successful. That is also usually asked. As you know, there is always a story behind a success or a failure. So, why he is so successful scientist? Now I'm going to tell. Uh, the first and the main reason he chose garden. The next point of Mendelian success was he take one character at a time. He studied, I can write, one or two. Next, he make isolated data for every experiment. In the next way, you can say he used some specific techniques which I told you earlier, like begging, emasculation. Next, he applied biostatistics that make his experiment error-free. And the last but not least, he chose all those characters which are found on different chromosomes. He was so lucky for this. Although Mendel was successful, but his work was not recognized at that time. Why? This one is also asked. The answer is the first one. His work was too complicated. As I told you, the data was not exactly 3:1. That was just like 2.9345 life. And these kind of calculations are quite typical for a biologist. You can understand this better. The second one, nobody was aware about a gene, genetics. So you can say the work was far away from that time. And the third and the last reason, but not the least, it's very important reason. As the Mendel work published, exactly at that time, the work of Darwin was also published as a book, Origin of Species. And he was a well known scientist at that time, so all the scientists were focused on his work. So nobody was interested about a new scientist and his work, and that's why he didn't get any fame for his work. But that was, you can say, that was his bad luck because he effort a lot, he work a lot, and very calculative work he has done. He has done a field work. It's a very typical task to calculate every flower bird, every flower experiment, as I told you. Suppose he was working on a field in which 1,000 plants are grown, and in a plant, 5 to 6 flowers are there, on which he is working in a different manner. Suppose in a flower he is discussing about or he is working about the pot shape, and in the other aspect, in other flower he is working on flower color. So it's a very typical work to manage by a single person. This one is managed by a single person. So you can realize that how calculated, how tough job he has done. But as we know, God is great. Then I can say exactly after his death, he died in he died on 6th Jan 1884. After the 16 years of his death, three scientists were there. Those recognized his work individually, and they were so honest because they gave this honor to Mendel. So you can see, now we will talk about rediscovery of Mendel's work. There were three scientists who were working on plants experiments in different countries, and ultimately they came to the same conclusion which was earlier given by Mendel. Now I'm going to tell about scientists. There was a Dutch scientist. First one was a Dutch scientist, Hugo de Vries. The second scientist was a German scientist, German botanist, called Correns. And the third scientist was Austrian botanist. He was Erich von Schumann. They rediscovered Mendelism. Now, what is exactly done by Mendel? How the monohybrid and dihybrid experiments are done? We will discuss it in our next lecture. Thank you.